Bismillahirrahmanirrahim and Assalamu alaikum. Dear students, our today's lesson is about the meiotic cell division. In our previous lesson, we have seen the detail of the mitotic cell division, and today we will discuss the details of the meiotic cell division. Most cells of the human body undergo cell division, the process by which the cell reproduce themselves. There are two types of cell division as we have discussed earlier. These are called the somatic cell division or the mitotic cell division, and the second type of cell division is called the reproductive cell division or the meiotic cell division. What is the somatic cell division? A cell undergoes a nuclear division called the mitosis, a cytoplasmic division called the cytokinesis. These two processes produce the two genetically identical cells, each with the same number and the kind of chromosomes as the original cell. The somatic cell division replaces dead or the injured cells and adds new ones during this tissue growth. While what about the reproductive cell division? It is the mechanism that produces gametes, the cells needed to form the next generation of sexually reproducing organisms. This process consists of the special two steps of division called the meiosis, in which the number of the chromosomes in the nucleus is reduced by half. And you can see the detail of this process by which that the meiosis is a form of the cell division by which the gametes with the half number of chromosomes are produced. Here the diploid number of chromosomes is reduced to the haploid number of chromosomes, and the diploid number is represented by the 2n while the haploid number is represented by the simple n. Meiosis is sexual reproduction. It is a two division process, meiosis I and meiosis II. Sex cells divide to produce the gametes, which are called the sperms or the eggs. Gametes have half of the chromosomes, occurs only in gonads. And you know what are the gonads? These are the testes and the ovaries. Male have the spermatogenesis process which produces the sperms, and the females have the oogenesis process which produces the eggs in them. Meiosis is similar to mitosis with some chromosomal differences. And what is the fertilization? A fusion of a sperm and egg to form a zygote. The zygote is a fertilized egg. This is the detail of the spermatogenesis process in which the human sex cells, which are the sex mother cells, which are actually 2n in number and containing the 46 number of chromosomes, are undergoing the meiosis process, first the meiosis I and the secondly meiosis II, and produces the haploid sperms. Now we will see all the stages of the meiosis I. Cell division that reduces the chromosomal number by 1/2. There are four phases of the meiosis I, which are called the Prophase I, Metaphase I, Anaphase I, and the Telophase I. Prophase I, it is the longest and the most complex phase, which accounts the 90% of the total process lapse in the process of meiosis. In this phase, the chromosomes condenses, synapsis occurs, in which the homologous chromosomes comes together to form the tetrad. Tetrad is the two chromosome or the four chromatids complex structure in which the sister and the non-sister chromatids are present. Here you're watching the process of the prophase I. So what happened in the prophase I? Here you're watching the process of synapse in which the two homologous chromosomes comes together and form a tetrad. Why it is called the tetrad? Because it is having the four chromatids at this stage. That's why this is called the tetrad complex. And what are the homologous chromosomes? So it's the pair of chromosomes from the maternal and the paternal partners that are similar in shape and size. Homologous pairs carries genes controlling the same inherited traits. Each locus is in the same portion on the homologous. Humans have 23 pairs of homologous chromosomes, 22 pairs of autosomes, and only one pair of sex chromosomes. What is the karyotype? A method of organizing the chromosomes of a cell in the relation to number, size, and the type. An important event of the Prophase I is the crossing over. What is the crossing over? The crossing over may occur between non-sister chromatids at chiasmata. Crossing over In the crossing over, segments of the non-sister chromatids breaks and reattach to the other chromatids. And what is the chiasmata? These are the sites of the crossing over. Here you can watch clearly the process of the crossing over. Now, after the prophase I, the second phase of meiosis I is called the metaphase I. It is the shortest phase in the meiosis. Tetrads align on the metaphase plate. Then there is the anaphase I phase. The members of each homologous pairs of the chromosomes separate as they are pulled to the opposite poles of the cells. The paired chromatids held by the centromere remain together. You're watching how the anaphase takes place. The last phase of the meiosis I is the telophase I. Each pole now the haploid cells of the chromosomes. Cytokinesis occurs and the two haploid daughter cells are formed, completing the meiosis I. This is the whole summary of the meiosis I process. After the meiosis I, the second stage of the meiosis started, which is the meiosis II. This is similar to mitosis. No interphase present or very short. No more DNA replication occurs before this. Meiosis II is similar to mitosis. Here in the Prophase II, during the early Prophase II, the chromatin fibers condenses and shorten into the chromosomes. The condensation process may prevent the entangling of the long DNA strands as they move during the mitosis. Then the metaphase II. During this phase, the microtubules of the mitotic spindle align the centromeres of the chromatids pairs at the exact center of the mitotic spindle. This midpoint region is called the metaphase plate. And during the anaphase, this phase, the centromere splits, separating the two members of each chromatid pairs, which move towards the opposite poles of the cell. Once separated, the chromatids are termed as chromosomes. As the chromosomes are pulled by the microtubules of the mitotic spindle during the anaphase, they appear V-shaped. And the last phase is called the telophase II, the final stage of the meiosis II. Telophase II begins after the chromosomal movement stops. The identical cells of the chromosomes, now at the opposite poles of the cells, uncoil and revert to the thread-like chromatin form. The nuclear envelope forms around each chromatin mass. Nucleoli reappear in the identical nuclei, and the mitotic spindle breaks up. So, this is all the detail of the meiosis process. Thank you and Allah Hafiz.